Welcome to the third video in the Translation and Protein Structure unit. In this video, we will discuss how translation occurs inside the cell. Translation requires many components in order to occur. Messenger RNA we have discussed previously. It is the template required for translation and is produced from the transcription of a gene. Translation also requires two other types of RNA molecules, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. It is important to note that all three types of RNA are produced by transcription of genes. However, only messenger RNA is then used as a template to produce proteins. Transfer RNA and ribosomal RNA genes are considered non-protein coding genes because the RNA molecule is the final product produced by the genetic information stored in those genes. Ribosomal RNA is a key component of the ribosome, the structure where translation takes place in the cell. Each ribosome contains a large subunit and a small subunit, which are made of both protein and ribosomal RNA. Interestingly, the protein functions to give the ribosome structure, while the ribosomal RNA is the actual catalytic component of the ribosome. It is the ribosomal RNA which is involved with forming the covalent peptide bonds to join one amino acid to the next during translation. The large subunit of the ribosome includes three pockets which provide binding sites for molecules of transfer RNA, as we will see when we look at the mechanism of translation. These sites are called the A site, or aminoacyl site, the P site, or peptidyl site, and the E site, or exit site. While it is the job of the ribosome to physically connect one amino acid to the next as the polypeptide chain is built, it is the transfer RNA molecules that bring the amino acid subunits to the ribosome and also function as the codon chart for the cell. While you have a chart you can read that says codon AUG should equal a methionine amino acid, the cell does not have a chart to read. Instead, it is the transfer RNA which properly read the codons and bring the correct amino acid to the ribosome. Each tRNA molecule folds up into an L-shaped structure pictured on the right due to various base pair interactions that happen within the RNA molecule itself. The two most important parts of the structure are the anticodon and the amino acid attachment site on the 3' end of the molecule. Transfer RNA molecules such as this one with no amino acid bound to the 3' end are called uncharged tRNAs. Before tRNAs can be used in translation, they must be charged. In other words, an amino acid must be attached to them. The enzymes required to charge a tRNA are called aminoacyl tRNA synthetases. Their role is to connect a specific amino acid to a specific transfer RNA molecule based on the anticodon nucleotide sequence. For example, the blue aminoacyl tRNA synthetase is unique to the amino acid isoleucine. There is a pocket in its tertiary structure that allows the isoleucine amino acid to bind. Additionally, there is a location in the tertiary structure of the protein that interacts with the specific UAG anticodon sequence. You should note that anticodon sequences are written 3' to 5' in order to properly complement base pair with the codon sequence in the messenger RNA. If you go back to look at the codon chart to find isoleucine, you will see that isoleucine here is encoded by 3 different codons. Each codon is written 5' to 3'. The anticodon region of the tRNA on the last slide was 3' UAG. Looking at the codons for isoleucine, we see that 3' UAG would form complementary base pairs with the mRNA codon 5' AUC. This is how tRNA molecules are able to be the codon chart for the cell. The tRNA synthetase enzymes link the correct amino acid to the transfer RNA based on its anticodon region, and the anticodon is then used to read the correct codons in the messenger RNA, similarly to how you read the codon in the chart and find the amino acid that it corresponds to. You should notice that there is a different tRNA synthetase enzyme for each amino acid. For example, the yellow tRNA synthetase binds to the amino acid glutamine, so it will charge tRNAs with the appropriate glutamine anticodon region, while the purple enzyme binds to the amino acid valine, so it would charge tRNAs with a specific valine anticodon region. Once charged, tRNAs are able to interact with the mRNA and the ribosome to bring the correct amino acids to build the specific primary structure of the protein. The elongation and termination stages of translation are very similar in prokaryotic and eukaryotic cells. However, there's a key difference in how translation is initiated between the two cell types, and that difference allows for different gene organizations. Genes in eukaryotic cells are considered monocistronic. Each gene contains its own promoter region and its own terminator region. Gene 1 and gene 2, each with their own promoter and their own terminator. This means that each gene is transcribed into a messenger RNA which encodes for only one protein. This is different than prokaryotic genes, which can be polycistronic. Polycistronic means that there are multiple genes controlled by the same promoter and the same terminator. In this case, gene 1 and gene 2 only have one promoter and one terminator. So the messenger RNA transcribed contains the code for two separate genes. This can occur in prokaryotes because of their mechanism of translation initiation. In eukaryotes, translation initiation occurs when the small ribosomal subunit binds to the 5' cap and then scans along the mRNA until it finds the AUG start codon. Translation elongation then continues until the stop codon is reached and the ribosome is removed from the RNA. If genes were organized as a polycistronic message, where two genes were controlled by the same promoter and the same terminator, that operon would be transcribed and processed into a polycistronic messenger RNA with a 5' cap and a poly A tail. However, when translation was initiated, the ribosome would bind to the cap, scan till it found the AUG start codon, continue translation until it found a stop codon, making a protein, and then falling off. Because the ribosome releases the messenger RNA when it gets to the UAGA stop codon, it would never make it to the second AUG start codon of the other gene. Thus, translation would never occur. The initiation at the 5' cap prevents the possibility of polycystonic genes in eukaryotic cells. However, in prokaryotes, the messenger RNA molecules have no 5' cap, so translation initiation must occur differently. Instead, the small ribosomal subunit binds to a specific nucleotide sequence of messenger RNA called the Shine-Delgarno sequence, seen here in this figure as the yellow boxes. Because Shine-Delgarno sequences are specific spellings of nucleotides, they can be found anywhere along the messenger RNA molecule. This allows for polycistronic gene organization in these cells, which is transcribed into a polycistronic messenger RNA message. In front of each gene is the Shine-Delgarno sequence, where the ribosome will bind, find the AUG start site, translate the gene, and then remove from the cell once it gets to the stop codon. At the same time, a second ribosome is able to bind to the Shine-Delgarno sequence in front of a second gene, scan till it finds the AUG start site, translate the gene until it gets to the stop codon, and be removed from the messenger RNA. In this way, the Shine-Dalgarno sequences allow each gene of the polycistronic message to be translated into a protein.